Hi everyone, welcome to the first video of Chapter 5, which is also the first video of Unit 3. This is, I think personally, this is where the class gets a lot more interesting, a lot more fun, because now we are at systems biology. Remember at the start of Unit 2, we started studying the actual living things, cells. Moved on into tissues, but now we're looking bigger. We are looking at body systems, organ systems. And the first system that we're going to look at is the integumentary system, which is the skin and all of its appendages, like hairs, glands, nails, things like that. So for the rest of this class and through Bio 139, we will be looking at much bigger pictures. We will be looking at body systems and in some cases, certain specific parts of body systems. So with that, let's go ahead and get started with our not really attendance questions. What is the function of stratified squamous epithelium? What are fibroblasts? And what is the most abundant protein in the body? Go ahead and try to answer those before listening to the answer. Hopefully we've done that. So let's see what the answer to those are. What is the function of stratified squamous epithelium? So first, let's think about what is stratified squamous epithelium. Stratified means there's more than one layer. Squamous means the cells are squashed or flat, elongated. So what's the function of stratified squamous epithelium? We find it in places that are exposed to the environment and friction. So stratified squamous epithelium is there to protect against abrasion. What are fibroblasts? Remember when we see BLAST at the end of a word, it means to build or to make. And usually it's building or making whatever's at the start of the word, fibers. So fibroblasts are cells that make fibers. We find these a lot in connective tissue where they make things like collagen or elastic fibers or reticular fibers, things like that. And what is the most abundant protein in the body? The most abundant protein in the body is collagen. And all three of these are going to come into play in today's lecture. So let's talk about the skin. The skin is the most easily seen part of the body, obviously. So it's the most easily seen system of the body. The skin and all the things associated with it is a system. And something that we might not often think about is the skin itself is an organ. The skin is the largest organ of the body. It's external, so we don't say it's the largest internal organ, but it is the largest organ. It typically weighs about 9 to 11 pounds in the average healthy adult white male. It makes up about 7% of the body mass. And it ranges anywhere from about 1.5 millimeters thick to 4 millimeters thick. The thinnest we find on the eyelids, the lips, parts of the genitals, and the thickest we find right between the shoulder blades and the back of the neck. Those areas have the thickest skin. And if we think about that, well, evolution, we came from animals that walk on all fours. So the back is up. The back is towards the sky. And that's the most exposed area. So we need really thick skin there for protection. And the skin is divided into a few different layers. The most superficial, or the outside layer, is called the epidermis, and it is avascular, meaning that it does not have a blood supply. There is no blood supply in the epidermis. Just deep to that, we have the dermis, So remember, epi means above or on the surface of or excess. So epidermis is above or on the surface of the dermis. So the dermis is just deep to the epidermis, and it is vascularized. It does have a blood supply. So let's think of something to kind of drive this point home. Let's say you get just a little bit of a paper cut, but it doesn't bleed. Well, that means you cut into the epidermis, but you did not cut deep enough to go into the dermis. Or let's say you get a little bit harsher of a paper cut or a little bit bigger of a cut in general, and it does start to bleed. You know you have cut all the way through the epidermis and at least into the dermis, because that's where the blood supply is. And lastly, we have the hypodermis. Now, the hypodermis is not a true layer of the skin, but it's so closely associated with the skin, they're kind of glumped together. The hypodermis is a layer of fat just deep to the skin. So we have epidermis on the surface, dermis just deep to that, and hypodermis just deep to that. So let's talk about the epidermis first. We will go through all of the skin layers, but let's talk first about the epidermis. The epidermis is stratified squamous epithelial tissue. So it's those many layers of flat cells. And there's a few different cell types there. Here over on the right, we can see an actual stained photomicrograph or a picture taken through a microscope. Over here on the right, we see an artist rendition of what we're seeing over here. So stratified squamous epithelium and different cell types. We have first keratinocytes. Most of the cells in the epidermis are keratinocytes. What are keratinocytes? Well, they are cells, sites. They are cells that make the protein keratin. Now, keratin is a protein that's kind of waxy. It's what gives your skin that kind of tough, waxy feel. It helps keep it waterproof. So it's protective. Next, we have melanocytes. Melanocytes are cells that make the protein melanin. Melanin is a pigment. It's one of the pigments that gives our skin its color. It's the primary contributor to skin color. We have dendritic cells. Dendritic cells, sometimes called Langerhans cells, they're these kind of branchy cells. They are, I like to say, they're kind of like the neighborhood watch. They are immune cells that are on the lookout for things that do not belong in the epidermis. And if they find something that doesn't belong in the epidermis, like a bacterium, they release these chemicals called cytokines or cytokines, and that calls the rest of your immune system to that area. Dendritic cells. And we have tactile cells. Tactile cells, sometimes in older books you will see them called Merkel cells. These work very closely with neurons or nerve endings. So think back to the tissue chapter. In the tissue chapter, we said that stratified squamous epithelium, in addition to being avascular, it also does not have a nerve supply. So here in the skin, same thing. There are no nerves running through the epidermis. They end at the dermis. But we need to get sensation to our nervous system. So one of the things that our body does is it has, setting right at the very base of the epidermis, we have these dendritic cells, and just deep to a dendritic cell, attached to the deep surface of it, is a nerve ending. So these Merkel cells will get information and pass it to this nerve ending, which will then send that information to our brain and spine. So that's how some information, things like chemical presence, things like that, that can be picked up by these tactile cells, these Merkel cells, and conveyed to the neurons that are in the dermis. But the epidermis itself has multiple layers. It is the most superficial layer of the skin, but itself has multiple layers. So starting from the deepest layer, I don't like to say bottom, but in this picture it is the lowest, the bottom layer. It is the deepest layer, and we're going to start there and work our way outward, or more superficial. The deepest layer of the epidermis is called the stratum basale. Sometimes you might see it called the stratum germinativum. I prefer stratum basale. That's the more common term, so that's what I will refer to it as. The stratum basale is a layer of stem cells. So what are stem cells? Stem cells are cells that are constantly dividing, and they will divide and produce two new cells. One of those cells remains a stem cell, and the other cell becomes something else. And then it can go through cell division. So here in the stratum basale, we have stem cells that produce keratinocytes, melanocytes, dendritic cells, tactile cells. So the basale, stratum basale, is dividing, and as it makes more, let's just focus on the keratinocytes because that's by far the most abundant. As it makes more keratinocytes, those keratinocytes are pushed upward. They are pushed more superficial. They move up into the next layer called the stratum spinosum. The stratum spinosum. The stratum spinosum is layers of keratinocytes. They're held together with desmosomes. Remember, anchoring junctions. Holds them tightly together but allows them to move a little bit. And they start making these keratin or pre-keratin filaments. So we're starting to get that waxy substance being produced. Meanwhile, the stratum basale is still dividing, still pushing more keratinocytes upward, more superficially. So cells that have been in the stratum spinosum for a while are now pushed even more superficially into the next layer, the stratum granulosum. Now let's think about something. Before we talk about the stratum granulosum, let's go back and talk about the epidermis and the dermis. The epidermis is avascular. It does not have a blood supply. The dermis is vascular. It does have a blood supply. So over here in this right image, we have blood supply down here, but it stops. So the further in this picture upward that we go, the further away we are from the blood supply, bringing nutrients and oxygen. By the time we get up to the stratum granulosum, we are pretty far away from the blood supply. So what happens? The stratum granulosum, these cells are starting to die off. When we look at them under a microscope, like we see here, they're a little bit darker. And that's because the organelles are breaking down. And as the organelles break down, they start to kind of wither. They're taking up less space. The cytoplasm is starting to get full of things like fats and keratin. And as they die, those lipids and keratin are going to be all that's left. So, meanwhile, stratum basale still dividing, still pushing cells up into the stratum spinosum, which pushes cells up into the stratum granulosum, which now is going to push those dead cells upward into the stratum corneum, the most superficial layer. So the stratum corneum, those cells are dead. They are pretty empty, but they're filled with keratin. And there's a lot of fat out in that extracellular space. Specifically, it's going to be things like glycolipids. Remember, fat with some sort of carbohydrate attached. So what happens? Well, by the time those cells are pushed all the way to the surface, it's flaked away. If you scratch your arm, you're flaking those cells away. If you put on clothing, you're flaking those cells away. If you take a shower, you are flaking those cells away. If you're just walking down the street, you're flaking those cells away. They're constantly getting flaked away. But that's okay, because we are constantly replacing them down here at the stratum basale. So they're constantly moving more superficial. And as they get to the most superficial, they're flaked away. And that's okay because they're dead. It protects those deeper living layers. So really, exfoliation, the act of removing dead skin, isn't all that great, because the purpose of those dead cells is to protect the living cells. So how do you remember the order of those? Well, come get some bacon. Everyone loves bacon. Everyone knows about bacon. Bacon will stick with you forever. So think now from the most superficial to the most deep. Stratum corneum, come. Granulosum, get. Spinosum, some. Basale, bacon. Come get some bacon. Corneum, granulosum, spinosum, basale. But we don't actually see it in this picture. There is another layer. Here between the stratum corneum and the stratum granulosum, there is something called the stratum lucidum. And the stratum lucidum is a clear layer of cells. They are mostly dead, just like the stratum granulosum, mostly dead. And in most of the body, they are so thin, the layer itself is so thin, we really don't see it under a microscope. Here in this image, we really can't see it. It would be right here, but we don't see it. The only places that it's thick enough to be seen are in the palms of the hands and the soles of the feet. Those two places, it's very, very thick and we can see it in a microscope, but most places we cannot. So let's look back at that shedding. We're shedding skin constantly. And as we shed skin, well, where does it go? What happens? Well, let's think about how much skin we're actually shedding, because, we don't really notice it. If you brush your arm, if your skin's pretty dry, you might see a little bit of it starting to flake away as this white stuff. That's kind of what dandruff is. There's a little bit more to dandruff than that, but it's kind of the basis for dandruff. Well, we shed about 50,000 cells per minute. And that's normal. That's not like someone who has an issue. That is normal to lose about 50,000 cells per minute, just skin. And over the course of your lifetime, that adds up to about 40 pounds. From the time you're born to the time you die, you have lost about 40 pounds of dead skin cells in your lifetime. That's a lot. But it's okay. Let's say you live in the same house your whole life. You don't see 40 pounds of skin laying there. No. Well, luckily for us, we live with little critters like dust mites and other types of mites and other types of insects like earwigs and stuff like that. And these little guys, they kind of creep along, too small to be seen most of the time, and they eat that skin. This is where it gets really fun. So they eat the skin, they take care of it. They're like our own personal little vacuums. Where do most of them live? Well, some of them will live like in the carpet, but most of them actually, they live in your mattress. And over the course of your mattress's lifetime, I know you're going to hate me for this, over the course of your mattress's lifetime, it gains about 10 pounds or so. What is that it's gaining? Well, some of it is dead skin cells. Some of it are dead dust mites, and some of it is dust mite poop. So yeah, your mattress is filling up with you and all your little critters that call you home. I'm sorry for that, but I always love to point it out just because we are much creepier than people think we are. All right, so now we're going a little deeper. We're at the dermis. We are now deep to the epidermis. And the dermis is mostly dense, irregular connective tissue. In lab, when we looked at the dense, irregular connective tissue slide, it was the skin slide. The stratified squamous epithelium, that was probably also the skin slide. We just looked at the superficial surface and the deep surface. So in this image up here at the top, this is the epidermis. Down deeper to that, so down at the bottom of this image, is the dermis. And the dermis has a few different cell types. We see fibroblasts. Remember, they're the cells that make fibers. Here, most of the fibers are collagen fibers. All of this kind of pink wavy stuff that we see here, that's collagen fibers. There are some elastic fibers here and there also. We will find macrophages. Macrophages are specific types of white blood cells that wander through our tissues looking for things to eat, either dead cells, dying cells, or invaders like bacteria or other debris. We'll find mast cells, another type of immune cell that's very similar to our Langerhans cell that we saw earlier. Mast cells are, again, like the neighborhood watch. They look for things that don't belong there. They release chemicals like histamine. If something shows up that shouldn't be there, that calls your immune system to the area. And then we see other types of leukocytes. So leukocytes are just white blood cells. Macrophages came from certain types of white blood cells, but we see other types there also, each serving their own purpose, but all there to protect you. And like we saw with the epidermis, the dermis is also divided into layers. It's only got 2 layers though. The more superficial layer is called the papillary layer. The papillary layer. And that's because it's got these kind of little raised bumps. If we see the pinker area, we see this kind of up and down, and then here another one a little wider that's up and down, and then we see the edge of one right here. Well, the papillary layer is like that. And each of those little raised bumps, that's called a dermal papilla. Papil means nipple, and that's just because of the shape of these guys. They're little bumps, so they resemble nipples. The dermal papilla, one of them. The papillary layer, all of them collectively. And the papillary layer extends back and forth up into the epidermis. So the epidermis and the papillary layer kind of zip together, kind of like a zipper, or like Velcro. They hold each other together. In the dermal papilla, we find a lot of collagen, a lot of elastin or elastic fibers. The papillary layer makes about 20% of the dermis. It's not very thick at all. We can see right here versus all of this. And those dermal papilla are highly vascularized. So not only do they have a blood supply, they have a lot of blood supply. They are full of capillaries and some small arterioles and venules. So there's a lot of blood supply there. That's why if you get a small paper cut that goes all the way through the dermis or through the epidermis, it will bleed a lot. Even if it doesn't do a lot of damage, it will still bleed a lot because the papillary layer is so vascularized. Now, in these dermal papillae, we will also find things called Meissner's corpuscles. Meissner's corpuscles, they are a specific type of nerve ending that are responsible for detecting light touch. So if you just barely touch the surface of your skin, you can still feel it, and that's because of those Meissner's corpuscles. It doesn't take a lot to feel it. On our fingers, our palms, our toes, and the soles of our feet, we will see fingerprints, footprints, handprints, and these are called friction ridges. Friction ridges. Here's one under a microscope. Friction ridges are primarily responsible for grip, especially if our hands are wet. So because it's not just completely smooth, because it's got this raised and sunken area, it allows us to grip things easier. And the shape, the pattern of these papillary, of the friction ridges is genetic. So identical twins or clones would have identical friction ridges, identical fingerprints, toe prints, footprints, handprints. But someone who's closely related, they won't have identical, but they will have similar patterns. Someone who's not related at all, their patterns will look very different. So here we see kind of these arches, but in some people it's more just kind of a wavy back and forth. So everyone, they will have sometimes subtle and sometimes dramatic differences. And in these friction ridges, we will often find things called lamellar corpuscles or Pacinian corpuscles. Lamellar corpuscles, they are very similar to the Meissner's corpuscles that we saw previously. They look very similar to each other. The lamellar corpuscles are a little deeper, but they are responsible for detecting things like vibration. And there's a lot of them in the friction ridges. Now we go deeper still. So we're still in the dermis, but now we are deep to the papillary layer. Now we're at the reticular layer, the thicker layer of the dermis. Makes up about 80% of the dermis. And it is still primarily dense, irregular connective tissue, but we will see pockets of adipose fat extending into it from the hypodermis. Remember, the hypodermis, not really a true skin layer. It's deep to the dermis. It's adipose tissue, but some of it will extend up into the reticular layer of the dermis. The dermis is primarily collagen fibers, and those fibers are kind of clustered together in bundles. And while they do run all different directions, the bundles tend to run roughly parallel to each other. And that's what these red lines are. These are called cleavage lines. So you can't see them just by looking at someone. But if we look at the bundles of collagen fibers, in the arms, they run roughly up and down. Same in the legs, roughly up and down. Around the torso and chest, they kind of form a ring around the body. There are some, bends here and there, but they all tend to run those directions. And that's important. If you're ever going to be some sort of surgeon, when we make incisions, if we cut parallel to the cleavage lines, when we sew them back up, there's going to be less gaping. They will heal quicker. They will scar less. So most of the time, if you look at someone who's had surgery, well, the cut is usually kind of parallel to these red lines. I had surgery on my neck to repair a slipped disc, and up here on the front, kind of on the side of my throat, there's a cut that goes kind of diagonal like that. If you know anyone who's had their appendix removed, usually the cut is kind of diagonal like this. One notable example is most C-sections are cut straight across the abdomen. I don't actually know why that is, but I've actually never seen one that does not go straight across the abdomen. Another thing that we find is called a flexure line. These are also or on the palms of the hand, the joints of the fingers, the joints of the toes, the back of the elbow, the back of the knee. So this is in places where we find repeated bending or flexing. So flexure lines, these are areas where the skin is being held very tightly to the deeper tissues. And this is, again, collagen fibers that cause this, but those collagen fibers are being used to very tightly hold the skin to the deeper layers. That way, during flexing, the skin doesn't kind of bunch up. It folds, it stays in place, and it keeps the skin from bunching up during flexing. All right, relatively short lecture today. That was just an introduction to the skin in general and some skin anatomy. In our next lecture, we will wrap up Chapter 5 by looking at skin physiology. We're going to look at things like skin color. We're going to look at a few different skin disorders. We're going to look at the different types of glands in the skin. We will not be discussing hair or nails in this class. They are in the chapter if you're interested in reading about them, but you will not be responsible for knowing about them for our class. All right, I hope you enjoyed the lecture, this first bit of coverage of a body system, and I will talk to you in the next lecture.